everyone, my name is Kirsti and I'm going to be talking to you today about giant guitarfish with a small emphasis on the species I work with as part of my master's research, the black-chinked guitarfish, and one of my sampling locations, the beautiful Santa Lucia Island in Cabo Verde, Western Africa, which you can see here as the background photo. But before we start, I just want to give a big shout out to the institutions and NGOs involved in supporting this work, including Sharks of the Atlantic Research and Conservation Center, Biosfera in Cabo Verde, the Save Our Seas Foundation, along with the University of Bremen and the Leibniz Center for Tropical Marine Research for having me as a master's student. I also wanted to thank Elasma Week for having me, and if you want to follow along on my journey or reach out to me at any time, you can find me on Twitter, username shown here. Alright, and just a little about me before diving into things. I grew up in a small town in Nova Scotia on the very east coast of Canada, where I was very fortunate to be surrounded by the ocean. As a kid, I was obsessed with all animals, especially those I could find while exploring the beaches, but my parents have told me that I've always had a particular love of sharks, as illustrated by one of my earlier artistic renderings dating back to 2002, when I was only 10 years old. Upon graduating high school, I stayed in Nova Scotia to do my bachelor's degree in marine biology at Dalhousie University, where I was a student in their first ever shark class. And then, after my degree, I decided to take a break from academia and move to the island of St. Martin in the Caribbean to work as a dive master, where I brought my passion of sharks with me, working with my bosses to help develop Patty's shark awareness course at their dive shop. Now, 6 years after graduating with a degree in marine biology, I am currently back in academia and halfway through my master's degree at the University of Bremen in Germany, where I've teamed up with the Leibniz Center for Tropical Marine Research and the Fish Ecology and Evolution Working Group to study the population genomics of black-chinked guitarfish in Cabo Verde. I have also come full circle, and this summer, 8 years after taking Dalhousie University's first ever shark class as a student, I was the primary lecturer for the course, getting the privilege to teach the students about what I love and take them all out blue shark tagging off the coast of Nova Scotia, just like when I was a student taking the class. During these tagging trips, we surgically implant acoustic tags that emit unique identifying pings, and when the shark swims past a receiver set up underwater that records these unique signals, the individual can be identified. In this way, we can track the movements of the tagged sharks and help identify when blue sharks are inhabiting Nova Scotian waters. In turn, this data can be used to help implement appropriate management and conservation practices, which is particularly important given that blue sharks are the most heavily fished sharks in the world due to direct-to-catches and bycatch, meaning that the species was not targeted but instead caught while fishing for other species. Essentially, this project, as a collaboration between Dalhousie University and the Ocean Tracking Network, seeks to identify if the same individuals are returning year after year, in which case Nova Scotian waters could prove to be a very important habitat for the North Atlantic blue shark population. But now, if we backtrack a bit, 2 years after graduating with my degree in marine biology, and at this point back from the Caribbean and once again in Nova Scotia, I found myself on the board of members of Canadian NGO Sharks of the Atlantic Research and Conservation Center, or SHARC for short, and with them participating in a research expedition that took our team to the Cabo Verde Islands. It is here where my story of the giant guitarfish begins, more specifically on the uninhabited island of Santa Lucia within the archipelago. And as you can see here on the map, where the group of islands can be found off the west coast of Africa, pretty much straight out from Senegal, and the black arrow on the map shows where within the archipelago Santa Lucia is located. The Cabo Verde archipelago, 500 kilometers off the coast of Senegal, Western Africa, is a former Serengeti of shark and skate diversity and abundance, hosting several endemic species that are species which are native to only a single geographic location and found nowhere else on the planet, and is home to numerous endangered species. All in all, Cabo Verde is a hotspot for sharks and rays in West Africa and the Atlantic. In 2017, our team traveled to Cabo Verde to tag Atlantic weasel sharks, one such endemic species, found only off Western Africa and the Mediterranean, including heavily fished and overfished regions. I'm sure this is a species most people have never heard of, so I promise I will come back to them, but I first want to draw your attention to the image here showing the global patterns in total fisheries catches from 1950 to 2014. The authors here were looking to detect ecosystem overfishing, and without getting too complicated, I just wanted to highlight what they found for the Cabo Verde region and just really ask you to keep these images in the back of your mind throughout the rest of my talk. You can see with the zoomed in images on the right, the increase in total fisheries catches around the Cabo Verde Islands from 1950 to 2014. And as of only yesterday, September 6th, a new publication by Nick Dolby and others was released showing that 1/3 of all sharks and rays are being driven toward extinction due to overfishing, with a disproportionate threat of extinction in the tropics, including the Cabo Verde Islands, where you can see on the map is highlighted in red, indicating that 75% of shark and ray species are threatened per cell in that area. I've pinpointed the Cabo Verde Islands here with the black arrow. And now, as promised, here is the Atlantic weasel shark, a globally endangered species getting no bigger than 1.5 meters fully grown, which you can visualize here if you use the hand and the pictures of the weasel sharks being released after tagging as reference. What's super unique about this species are the purple and yellow stripes that run along the length of the body, which you can see best in that last picture, in particular those purple stripes. I also wanted to highlight here that the Atlantic weasel sharks are a good example of what many sharks look like. Well, maybe not the purple and yellow stripes, but size-wise, many species of sharks on the planet only grow to maximum lengths of 1 meter or so. Yet global media focus seems to emphasize the bigger sharks, like white sharks and tiger sharks, still fixating on fear-mongering images and stories, which finally brings me to my introduction of giant guitarfish. I've been intentionally leaving out pictures of these species until now because they are so incredibly unique looking, and again, not at all what most people typically think of when talking about sharks and rays. And without further ado, meet the giant guitarfish. More specifically, here we have the black-chinned guitarfish, this photo of which comes from our project partners at NGO Biosfera in Cabo Verde. These species are shark-like rays, named for their wedge or spade-like body shape resembling the shape of a guitar. These species are shark-like because their muscular tails are more similar to that of sharks versus the longer and slimmer tails of rays. This tail, paired with the large dorsal and caudal fins, are perfect for providing quick propulsions and easy maneuverability because they are more muscular than the typical ray tail. These species are also ray-like, as you can immediately see from their flattened head and body with an elongated triangular snout that allows the species to bury more easily into sandy bottom environments, either in search for prey or to camouflage itself. And aside from the obvious morphological features such as the body shape, one cool feature that differs between sharks and giant guitarfishes are their eyes, more specifically how they protect their eyes, which is shown really nicely in the following short video.
Creatures like the giant guitarfish, a type of ray that gets its name from its guitar-shaped body, can't blink. Instead, they retract their eyeballs, pulling the organs almost completely into their heads. Now, new research shows the fish have that ability thanks to a special eye muscle. The same muscle in sharks, who don't retract their eyes, is smaller and attaches the eye to the middle of the eye socket. But in guitarfish, the muscle is larger and longer. It connects the eye to the underside of the skull, giving the creatures an increased range of motion. That helps the animal protect their eyes, like our eyelids do when we blink. Just don't challenge these tropical fish to a staring contest. You're sure to lose.
Okay, it's pretty awesome, but how do we classify the different species of giant guitarfishes? Well, First, we have to know the order they fall under. To differentiate between species, plants, animals, bacteria, fungi, and protists are taxonomically classified using this 7-step Linnaeus system, and it's no different for classifying the different species of giant guitarfishes. But to give you an example, your pet dog falls under the kingdom Animalia for animals versus plants, for example, under the phylum Chordata and class Mammalia, which, as the name suggests, is where you would find all mammals. The order Carnivora, animals specialized in eating meat, family Canidae, which are dog-like carnivores, the genus Canis, which includes extant or living dog species including wolves, coyotes, jackals, and domestic dogs, and finally the species name would be Canis familiaris. And, like my parents' pet duck-tolling retriever, giant guitarfishes also fall under the kingdom Animalia and phylum Chordata, but because giant guitarfishes are fish and not mammals, their taxonomy differs at the class level, falling under the class Chondrichthyes, which includes all the cartilaginous fish species, sharks, skates, rays, and chimeras, all of which have skeletons consisting primarily of cartilage, the same flexible tissue we have in our ears. Giant guitarfishes then fall under the order Rhinopristiformes, or more simply, shovel-nose rays. And within the order of shovel-nose rays, there are 5 families: banjo rays, sawfishes, wedgefishes, guitarfishes, and giant guitarfishes. So here they are, the shovel-nose rays. But what I want to highlight here is just how similar the guitarfishes, giant guitarfishes, and wedgefishes look. In fact, guitarfishes and giant guitarfishes used to be grouped together into a single family. However, molecular analysis, so looking at the DNA or genetic makeup of these animals, has found that the giant guitarfishes are actually more similar to sawfishes than they are to the family of guitarfishes. Hence the separation into 2 separate families, guitarfishes and giant guitarfishes. Confusing, I know. So how do we tell them apart? Well, wedgefishes have spots or ocelli, a fancy word for eye spots, and have a lower caudal lobe, which is the lower lobe on their tail fin right here. Guitarfish nostrils are rounded with their nasal openings either circular or oval in shape, and there is no sharp demarcation between the snout and the cranial cartilage, which is a fancy way of saying that there is no sharp line separating the rostrum or the nose of the guitarfish from the head. Whereas giant guitarfish, as you can see on the other hand, have a sharply demarcated snout here and cranial cartilage, which is a clear separation between the nose and the head. They have long, narrow nostrils with rectangular shaped openings and typically do not have spots or ocelli. However, both guitarfishes and giant guitarfishes lack that lower lobe on their tail fin that wedgefishes have, which make distinguishing between guitarfishes and giant guitarfishes the bigger challenge. But if you take a look at those nostrils, you can get a better idea of the rounded versus rectangular shaped nasal openings that can help in identification, which is something that will come up again when I talk about how we identified the black-chinned guitarfish, a species of giant guitarfish in Cabo Verde. So again, jumping back to our Cape Verde expedition in 2017, knowing these features became key in helping us determine the species we were working with. While on the field expedition to tag Atlantic weasel sharks at the uninhabited island of Santa Lucia, our team was the first to scientifically validate the presence of the black-chinned guitarfish in Cabo Verde. Here you see a picture taken from that trip where I'm releasing the guitarfish after we had taken measurements, pictures, and tagged the individual using external T-bar tags. These T-bar tags, named from their shape, which you can see in the picture on the right, are inserted into the musculature next to the dorsal fin, anchored by the T. The tags have a unique number and contact information on them, and they are used in mark recapture studies. For example, when the animal is first tagged, the tag number, along with information such as length and date of capture, is recorded onto a data sheet. If the animal is then recaptured, the length and the date of the recapture are recorded and the individual is identified through the unique tag number, and then compared to the initial tagging data. In this way, we can see how fast the animal grew, and this growth information can then be used to help appropriately manage the species, because knowing the growth rate is an important factor in determining longevity and the mortality rate of a species. But how did we determine that the species we caught was indeed a black-chinned guitarfish, when differentiating between giant guitarfishes and guitarfishes is so hard to do? Well, we measured the shark. Typically, giant guitarfishes are larger than guitarfishes. We photographed the nasal openings and saw that they were longer and more rectangular in shape, another indication that we were looking at a species of giant guitarfish, and then checked habitat ranges for all giant guitarfishes, and concluded that only the black-chinned guitarfish had the same morphological features as the species we were seeing and could be found off Western Africa. We then worked together with the local taxonomic specialist and professor at the University of Cape Verde, Rui Freitas, to include this first scientific confirmation of the black-chinned guitarfish in Cabo Verde in his 2018 publication on new and confirmed records of fishes from the Cabo Verde archipelago based on photographic and genetic data. Notably, black-chinned guitarfish are named because some individuals, in particular newborns and juveniles, have a black patch on their chins. However, this patch can fade as the individuals reach maturity, but you can see a nice example of this patch from a baby born into the National Sea Life Centre in the UK as part of their breeding program. Since the 2017 expedition, Biosphere has continued to travel to Santa Lucia throughout the year to tag and release more black-chinned guitarfish. Here's a short clip from a video their team made showcasing their tagging efforts. The subtitles in the video are in Portuguese, so I will translate them as they come up. Shark and Ray Tagging Expedition. Santa Lucia, Cabo Verde. Landing, setting up the camp, and preparing for the capture and tagging activity. We start at dusk or dawn. Capture is done in the style of surf casting or beach fishing with or without bait, using barbless hooks. The target species, Glaucostegus simiculus, or the black-chinned guitarfish. Once captured, we have a maximum of 90 seconds to remove the hook, measure, and place the T-anchor tag with a unique number per individual. Take DNA samples from the loose part of the pectoral fin and store properly. Ensuring good handling with minimal stress and subsequent release into the sea. And the process repeats itself. Always ensuring good handling and that it is a maximum of 90 seconds. This brings me to the topic of my master's thesis, genomic analysis and effective population size of the critically endangered black-chinned guitarfish in Cabo Verde. I know, the title is a mouthful, but don't worry, I won't get into discussing anything too complicated at this time, but instead introduce you to the importance of and the goals of this project. So, why is this project important? Well, as it turns out, of all fishes in the ocean, giant guitarfishes and wedgefishes are the most at-risk group of marine fish globally. These species are medium to large shark-like rays, as discussed before, occupying shallow coastal waters throughout the Indo-Pacific and Eastern Atlantic Oceans and the Mediterranean Sea, unfortunately overlapping in areas of high and increasing fishing pressures. Remember the map I showed you toward the beginning of the presentation? The vulnerability of these species is also increased due to their limited distributions and having low biological productivity. From what we know, both wedgefishes and giant guitarfishes give birth to only a small number of young at a time. However, we are still lacking reproductive data for 6 of the 10 species of wedgefishes and 2 of the 6 species of giant guitarfishes. Furthermore, it is not known whether or not these species give birth annually, although it is suspected that black-chinked guitarfish undergo annual reproductive events. Additionally, giant guitarfishes and wedgefishes have the most valuable fins in the fin trade, and they are targeted for their meat and coastal subsistence fisheries. Guitarfishes and giant guitarfishes have even been used as art and are considered by some as the earliest fake mermaids. These sculptures, if you will, are called Jenny Hanivers, and the pictures here are of those that have been showcased in the UK Natural History Museum. In fact, the earliest known record of a Jenny Haniver dates all the way back to 1558 in Historia Animalium, Volume 4, a book by Swiss physician, naturalist, bibliographer, and philologist Konrad Gessner, who wrote that these figures should not be mistaken for mini dragons or monsters, which was what was believed by some at the time. So, now we know that wedgefishes and giant guitarfishes are targeted for their high-value fins, occupy habitats that overlap with high and increasing fishing pressures, and have even been fished for art. But what does this all mean in terms of population trends? In a 2020 study by Peter Kine and others, published in the journal Aquatic Conservation, the authors applied the IUCN Red List criteria and assessed all but one of the 16 species of wedgefishes and giant guitarfishes as critically endangered. The IUCN, or International Union for the Conservation of Nature, bases this criteria on the geographic range of the species, population size and the rate of increase or decrease, along with an analysis on the probability of extinction. On the bottom, you see a figure from this study, where the black line shows the global Red List index for these species since 1980, where a Red List index value of 1 indicates that all species are categorized as least concern, and a Red List index value of 0 indicates that all species have gone extinct. Yet, despite the critically endangered status of these species, Peter Kine and co-authors emphasize that there is still comparatively little available information for giant guitarfishes and wedgefishes. In short, a critically endangered status on the IUCN Red List means that a species is at extremely high risk of going extinct in the wild. The project that I am working on as my master's thesis will help fill critical gaps in knowledge for one of the species of giant guitarfishes, you guessed it, the black-chinked guitarfish. This species can be found in waters up to 100 meters and is distributed throughout the eastern Atlantic Ocean from Angola up to Portugal and including the Mediterranean. The black-chinked guitarfish is assessed as critically endangered and has decreased in population size by over 80% in just the past 3 generations or 45 years. And yet critical gaps in knowledge for the species remain, including lack of population status data, lack of reliable longevity estimates paired with the minimally available genetic and genomic data for all giant guitarfishes, wedgefishes, and guitarfishes more generally. To fill these gaps in knowledge, the objectives of my work are as follows. First, to assemble a reference genome for the black-chinked guitarfish in particular, and for the group giant guitarfishes more generally. And second, to estimate the effective population size for black-chinked guitarfish in Cabo Verde. But what is a reference genome, and what is effective population size? Well, a little bit of a warning, the next few slides might seem a little overwhelming, but they're really just scratching the surface on reference genomes and effective population size, and how they can both be used in species conservation. But it really helps tell the story of my project, so just bear with me, and I will try to explain these terms as simply as possible. So, a reference genome is a digitalized nucleic acid sequence database that can be considered a representative and idealized set of genes or all the genetic information for the species of interest. Currently, there are no reference genomes for any of the giant guitarfish species. However, reference genomes are an important resource in endangered species conservation by providing means to evaluate demographic parameters such as diversity, longevity, patterns of inbreeding, hybridization and introgression, disease susceptibility, and adaptation. Furthermore, my thesis work will involve using such a reference genome to further evaluate the effective population size of black-chinned guitarfish in Cabo Verde. However, this reference genome will also be an important resource for the group of giant guitarfishes more generally, since there is an overlap in genetic makeup in these related species. For Objective 2 of this project, where I look to estimate the effective population size for black-chinned guitarfish in Cabo Verde, I have teamed up with local NGO Biosphere, who I've worked with in the past when I was part of the team that first confirmed the presence of black-chinned guitarfish in Cabo Verde. Specifically, we are looking to estimate the effective population size for the population around Santa Lucia Island, where the species was first validated back in 2017. Shown here is a map of Santa Lucia Island within the Cabo Verde archipelago and relative to its location about 500 kilometers off the coast of Senegal, showing the number of black-chinned guitarfish caught and released as part of ongoing shark and ray conservation work by Biosphere, and compared to other shark and ray species caught at sampling location 5, illustrated as the red dot on the southwestern bay of the island. Black-chinned guitarfish make up the clear majority of the species being captured as part of their tagging program, shown as the highest bar on the graph, with 83 different individuals captured, tagged, and released since 2019. During the tagging process, small fin clips, less than 5 millimeters in size, are taken from the trailing edge of the pectoral fin and preserved so that I can analyze the DNA of this population. Importantly, the Cabo Verde archipelago has been quoted as a hotspot for sharks and rays in West Africa, as I said before. However, a 2019 paper from Rui Freitas, also as mentioned before, an instructor at the University of Cabo Verde, discusses increased fishing pressures within the island nation, including places like the here uninhabited island of Santa Lucia, despite being a marine reserve. My thesis project will therefore estimate the effective population size of the Santa Lucia population of black-chinned guitarfish to help inform appropriate long-term management and conservation practices in Cabo Verde, as results from this project will be shared with the National Director of the Environment as a condition of my research permit. The effective population size is used to predict the rate of inbreeding and loss of genetic variation in a population, whereby a loss in genetic variation could decrease the fitness of a population and reduce the population's resilience to pressures such as environmental changes. An effective population size greater than 1,000 is needed to maintain fitness of a population into perpetuity. I hypothesize that the effective population size of the black-chinned guitarfish from Santa Lucia will unfortunately be less than 1,000, which would indicate that this population would not be able to maintain its fitness into perpetuity. I am basing this hypothesis on the inferred isolation of the species. Water depths between islands in the Cabo Verde archipelago are often greater than 1,500 meters, which exceeds the 100-meter coastal habitat range that black-chinned guitarfish are known to occupy. And therefore, these water depths could act as a dispersal barrier, isolating the black-chinned guitarfish population in Santa Lucia from the rest of the archipelago and mainland Africa, and restricting migrant gene flow, because a population from one area could not reproduce with individuals from another population, ultimately reducing heterozygosity, which reduces the genetic variation, and as mentioned earlier, could then reduce the fitness of a population, and thus reducing the resilience of the population as a whole. If my hypothesis holds true, it would indicate the need for increased management within the Santa Lucia Marine Reserve, which is why I'm working together with Biosphere and Cabo Verde to ensure there is sufficient information about the giant guitarfish within the island nation that could then be used to establish appropriate management and conservation practices. And now, Before wrapping up my presentation, I also just wanted to highlight some of the other species of giant guitarfishes, wedgefishes, and guitarfishes, since I've primarily focused on the black-chinned guitarfish here. But these are such weird and wonderful animals, I wanted to share a few more with you, such as the club-nosed guitarfish shown here, a species of giant guitarfish found in the Indo-West Pacific, along with a record from the Red Sea. This species is easily distinguishable from all other giant guitarfishes and guitarfishes because of its bulbous nose tip. And how about the false shark ray, a species of wedgefish, possibly restricted to the coastal waters off Mauritania, Western Africa, making it an endemic species if so, occurring nowhere else in the world. Unlike other wedgefishes, giant guitarfishes, and guitarfishes, you can see here the false shark ray has a blunt nose. But not to be confused with the shark ray, shown here, which has a much wider distribution and can be found in the Indo-West Pacific, South Africa, New Caledonia, and all the way up to Japan. Aside from geographical differences, the shark ray differs from the false ray with its broader head and ridges with more prominent large thorns along its back, shoulders, and above the eyes. Or how about the Gorgona guitarfish, a species of guitarfish with particularly large eyes and covered in small white spots, found from northern Peru up to Mexico. And lastly, as of this summer, a newly described species of giant guitarfish, scientifically named Glaucosteges yonholei, was found within the Bay of Bengal in the Indian Ocean, just really highlighting how little we actually know about these species. So little, in fact, that we're discovering species we never even knew existed prior to this summer. And now that we're nearing the end of my talk, I wanted to leave you all with this in mind. The relationship between sharks and humans has always been a bumpy one, dating back centuries where explorers and fishers feared them. But what I think is not discussed enough is that the relationship between skates and rays and humans has also been quite negative in the past, an example of which can be seen here from the Macclesfield Psalter, dating back to the early 1300s, where the picture is described as a skate terrorizing a man. The intention throughout the manuscript was to illustrate grotesque images, giving you an idea of what people thought of skates and rays hundreds of years ago, fearing them. While in reality, we should be more fearful about losing them. And to end, I'll leave you all with this beautiful video made by our partners in Cabo Verde at NGO Biosfera, where you can see not only the black-chinned guitarfish I work with, but some of the other incredible wildlife, including other sharks and rays. Again, the subtitles in the video are in Portuguese, so I'll translate along as we watch. They've been here for over 400 million years. Sharks and rays, a heritage to be preserved. There are more than 1,000 known species. With different shapes and sizes. Their presence is essential in the marine ecosystem. Top predators contribute to greater biodiversity, influence the behavior of other species, leading to the creation of schools of fish as a form of defense, and contribute to the control and health of our seas. They feed on the weakest and sickest fish, preventing the spread of disease in the oceans. However, they are vulnerable to overfishing due to their low recovery capacity. Therefore, protect the sharks and rays. It's protecting our own oceans. It's protecting what's ours. According to the IUCN Global Red List of endangered species, approximately 60% of the pelagic sharks are currently in danger of extinction. Again, I want to thank my project partners at Sharks of the Atlantic Research and Conservation Center and Biosphere for helping to make our field expeditions possible, the Save Our Seas Foundation for their support in the project, and the institutions through which I'm completing my master's thesis, the University of Bremen and the Leibniz Center for Tropical Marine Research in Bremen, Germany, and under the supervision of Professor Oscar Puebla. And lastly, I want to give a big thank you shout out to the folks at Elasma Week for having me, and a big thank you all for listening and watching along. If you have any questions, again, please don't hesitate to reach out to me. My Twitter handle is @Kirsti_Burnett, and I hope to be talking to some of you soon. Thank you.